Greetings, I'm Salvador Cordova, and welcome to the Evidence and Reasons for the Christian Faith video channel. This is just going to be, this show is just going to be a free-form discussion with Jeremy, and I'm privileged to be here with Jeremy as a co-host and guest. So, Jeremy, tell me what's on your mind and what you'd like to talk about today.
Well, after a busy day, and after our previous communications over doing this, I'm really stoked about the topic of the biotic message.
Can you, if I do this, that means bring the microphone a little closer to you.
Oh, yeah?
Okay.
All right.
Good.
So I am really interested in sort of the history of the Biotic Message book and you, since you have a unique history concerning it. My history with the book is when I was going to university, I joined InterVarsity Christian Fellowship, and I also joined, what was that other one? I can't remember, but there was a friend from the other one. It's the other big university outreach. And this man started reading the book and was just enthralled, and he bought a big box of them. And he said, "Do you want to buy some and give some to your friends?" And I said, "Sure." So I bought about 7 of them. And we started having meetings based on the Biotic Message, about 6 or 7 people around a table and talking about the amazing content of the book. At that time, it was groundbreaking, and so I started reading it. I found it very difficult past chapter 4, but I was amazed to see on the CBC, Canada's national public broadcast, that they had a 2-hour special dedicated to the book. Because I was familiar with the book at that point enough to know that this is what this whole thing was about. And they had a simulcast from multiple universities and audiences posing questions, trying to answer questions concerning the questions raised by this book. So I think it's extremely significant book, The Biotic Message, and it changed my understanding of the debate, because up to that point, the way I discussed the topic was through induction, and I was getting completely worn out by trying to present more and more evidence. It just seemed to be an endless array of tit for tat for all these, and I couldn't really get into deeper topics at that point. I was just a computer science student and didn't have physics or biology in my background, or paleontology. But I had read a few of the Creationist books that were put out by Answers in Genesis, and a number of websites. And I had enough of a understanding of the facts that I could discuss it, but it was hard to keep from getting emotional, because it was just such a pivotal discussion. So much swings back and forth, based on the evidence you present. So when I started reading the Biotic Message, I was very relieved that there was another approach that you could take concerning the topic, and that was through deduction. Analyzing the philosophy of the theory of evolution, the philosophy of science, and understanding the logical fallacies that are introduced by different aspects of the evolutionary theory. So it was very important to me that way, because it really allowed me to sit back a little bit and just discuss ideas rather than present one item of evidence one after the other, which was wearing me completely out and stressing me out. So that's my background.
I'm actually not that familiar. There is a difference between deduction and induction, and unfortunately, I'm not that acquainted with it. Now, if you saw me, for the audience, if they saw me looking this way, I have like 4 computer screens in front of me. I was desperately looking for a PDF of the Biotic Message. You can find it on the internet. I think, I'm not 100% sure if Walter authorized this, but I do have a copy of it somewhere in my house. So I felt this may be, may end up actually being good, because we could actually talk about some of the pages and we could at least look at the book chapter. So let me unshare my screen while I desperately try to get this on my other computer here. And that was very interesting. Can you, our mutual acquaintance, Danny, the philosophy teacher, he'd probably get on my case to give me the distinction between, what was it, induction and
deduction.
So you were saying that induction was very frustrating for you. Can you tell me why that is?
Induction means the input of evidence. Deduction means the deconstruction of evidence, or the trying to understand the evidence. And I guess the logic proposed in regards to the evidence. So you're not looking at the evidence necessarily completely, you're looking at the logic that surrounds the evidence.
Okay. So, so actually this may be a real big educational session for me. You can make the distinction. So you're saying prior to reading the Biotic Message, what sort of arguments were you involved in? Did you use resources from other
I was looking at websites. This would have been my initial concern started in school, and when I first encountered the theory of evolution, I said, this doesn't sound like Genesis. And I was always putting up an argument in class and almost getting in trouble for doing so. And I remember one day someone presented an argument to me that I couldn't answer, and just in a sort of a last-ditch attempt to save my position, I just said, "Oh, that's just a story." No real deduction there. But the problem was, whenever I started to argue in class from that point on, someone would pipe up and say, "Oh, Jeremy, that's just a story." I never lived it down.
Wow.
But once I started getting into, the different ways you can argue it, you can, the fossil, the strata, the cosmology, the astronomy, the different things like, the dust layer on the moon. I was aware of it, about that at the time, and how, before they went to the moon, they estimated that that there could possibly be like 190 feet of dust on the moon based on the age of the dust and the annual downfall of dust. So when the footprint only made 1/8 of an inch in the moon sand, that was very telling and revealing. So it was a massive contradiction. So I think I started to make a pivot when I started realizing how genetics was variable within the same kind of creature, like the peppered moth. The idea that you have a population where there's a spread of white and black moths, and depending upon the environment, if there's a change in the visibility of the moth, you'll have a expression or an increase in the moth that sort of hides within that color scheme. So that was very interesting. I couldn't really get into that much because I wasn't much into reading beyond a certain level of detail, because it was very intimidating, I found, to open the textbooks and to get into the chemistry, get into the biology, get into the genetics. So once I got into the philosophy of science, the philosophy of evolution, the philosophy of what the Bible says, then I started having more in-depth discussions, trying to get more control over my emotions as I discussed the topic, because I was so anxious. It just made me extremely anxious to talk about. But I was so I gravitated to it. It's like catch-22. I couldn't help myself. I gravitated to this thing that made me miserable, basically, that I wasn't really prepared to, discuss in any meaningful way. But I got to the point where I actually convinced a few people. They were evolutionists, and they became born-again Christians, young Earth creationists.
Wow.
Yeah,
Wow.
so
Well, that's, thank you for sharing that. Now, interestingly, I asked Walter, I think he probably wouldn't mind me sharing this. I asked him, why did you even get involved in this? And it's like a lot of people, they're just searching for meaning. And he was searching for meaning in life, searching for God, and that question came up. So I'm very grateful that you have raised this topic. Walter ReMine's work in this book, this was his life work. And the influence it had on my journey was that I was nearly leaving the Christian faith in 2000. This book was published, I think one of the publication dates was 1993. 1993, there it is. And so I got a hold of the book, say around the 2001 time frame, and I was just grabbing every book I could, good and bad. But this one really struck me. It had a different tone. Unlike any other creationist book I started with, this didn't mention the Bible at all. It shocked me. And I said, I actually like that. And the reason is that if one is having doubts about the authenticity of the Bible, that means you can't use it as a starting point anymore. And many years later, I'd read the verse in John 10:38. Jesus said, if you do not believe me, believe the works. And Blaise Pascal also later argued that that's saying that he said the Christian faith is established by miracles, not just mere words. And I found that very compelling, that Jesus is not saying that you have to start with the right worldview to arrive at the truth. In fact, if that were the case, I wonder how many people would actually change their minds. He gives, he acknowledges the situation where maybe someone isn't arriving at the, is not starting his search with the right worldviews. But then the evidence kind of just confronts you and gets you to change your mind. And in mathematics, we call that, kind of the formal parallel to that is proof by contradiction. You actually assume a certain argument and you realize it just can't hold up. Then you realize the opposite must be true. So for very binary type questions, you can assume one view, and if it is inconsistent, then you will assume the other view is correct. The famous proof of the square root of true being irrational is like probably the first case in mathematics to that effect. So I said, this is great. I can actually just start with the naturalistic worldview and just see where it leads. And if the naturalistic worldview doesn't explain life, the alternative is something supernatural or something like it. At the very least, that's where I began. And that's what really impressed me about the book. The other thing is it didn't address Noah's flood or the long ages? I said, yeah, I could, because I was a physics student. I'd been an old Earth creationist for a very long time, and I was revisiting these questions because I was having a lot of doubts about God and creation and everything. I wanted an approach that didn't put that in the forefront. Some of the stuff by Josh McDowell on the issue of the distant starlight and all, I just found very distasteful. And to my surprise, even Answers in Genesis didn't like McDowell's solution of created light, where we have all these events in the heavens that never happened. So some of the stuff that, not to diss all of Josh McDowell's work, some of this stuff on creation is not very persuasive to me. I found it actually distasteful. And it was refreshing to see that other creationists felt the same way. So I said, I'd rather begin with, okay, I'm not going to try to prove everything all at once. But if we can disprove abiogenesis, and if we can disprove evolutionary theory, creation starts to become very compelling, and it becomes easier to accept. And that's where I was. So that's why I found this compelling. And then he did go on some, he did explore some things that were just also very obscure, which I sort of like that, because I me, I'm a tremendous nerd, and so even if I don't understand something, I find it just compelling when he goes into these very complicated things like Haldane's dilemma and Fisher's fundamental theorem of natural selection. I found that really compelling.
Yeah, chapter 4 on natural selection is actually a retrospect of what actually took place in the 1800s from many scientists who were reviewing the theory, and it was such an eye-opener to see the power of correctly labeling that idea as a tautology, which is a definition that masquerades as higher knowledge but explains nothing. So once you get into that, that's really powerful in a conversation. I mean, you could have 24 pages of dissertation, but if it's formulated in a tautological form, you basically have something that explains nothing.
Exactly. So now that I'm looking at the chapters, and it's been a while since I've slugged through the book. I mean, this is like maybe 10, 15 years. Mm-hmm. But one could easily see that some of the themes you've seen in my discussions came from here. So like chapter 3, let me zoom in a little bit here because the font is a little. Yeah. Chapter 3, the origin of life. Now, I specifically talked to Walter. I said, that's our strongest case. He said, I was surprised you didn't. I said, Walter, I'm surprised you didn't cover it more. He said, no, we got that one down pat. The only reason I gave it, the main reason he gave it only one chapter, he said he knew we were going to win that argument. And that's played out, which is kind of prophetic in light of the book by Change Tan and Rob Stadler, the Stairway to Life. They pretty much demolished the abiogenesis argument. So yeah. Walter said I said, Walter, there's so much on this. He said, no, I'm not covering it because other people are going to cover it. We own this argument. We're not going to lose it. And
yeah, that's something that is specifically an inductive argument where you're actually going into all the mechanical aspects of what's being proposed.
Okay. Well, again, I don't know all the nuances, but maybe I'll learn along the way. And then now chapter 4, 5, and 6, survival of the fittest, inventive natural selection in Darwinian scenarios. Let me just read it since you said this was so influential. Chapters 4, 5, and 6, survival of the fittest, inventive natural selection in Darwinian scenarios, and an appendix cover much material. Yet they accurately reflect the great diversity of defenses that evolutionists have made for Darwin's theoretical mechanism. A lengthy treatment is necessary to document this controversy, which in its modern form has already evaded both clarity and resolution for many decades. Evolutionists claim that natural selection is a scientific explanation for life's biological design. These chapters dismantle that intricately crafted illusion. Oh my.
Yes. And it's interesting, he has a background in magic.
Yes, he's an
So
illusionist.
he said that really contributed to his viewpoint
I
of,
didn't. Wow.
understanding the sleight of mouth, basically, rather than the sleight of hand of evolutionary theory.
Then chapter 7, 8, 9, population genetics, Haldane's dilemma, and the neutral theory of evolution. I'm not going to go into the details here. Just chapter 10, gradual integrations in phylogeny. Chapter 11, modern systematic methods. Oh my goodness, that's hard.
Yeah.
Just for the reader's interest, if anyone is wondering, I think Walter ReMine said he's a transformed cladist. So people from the evolutionary schools would know what that would mean. I kind of know it and then unknow it. Evolutionary illusions shows how words and imagery are used to create the illusion of ancestry. Chapter 13, the fossil record. Chapter 14, punctuated equilibria. Chapter 15, hierarchy theory, shows how modern theorists have shifted away from phylogeny to life's nested hierarchy as the major prediction of evolution. This chapter dismantles the illusions created by punctuated equilibria, hierarchy theory, pluralism, and anti-reductionism. Now this is what, this is almost getting to be about 30 years ago. So there's some things that may have been that might need updating.
Mm-hmm.
Chapter 16, nested hierarchy and convergence, examines the pattern of life in morphology space. It shows that message theory predicts nested hierarchy and convergence, while evolution never did. Also, Darwin's riddle is cleanly solved. This is a key chapter, though it cannot be fully appreciated without the groundwork laid earlier. Chapter 17, embryology, explains this classic body of evidence, especially von Bayer's law of development. I'll move on. Chapter 18, vestigial organs. Chapter 19, molecular evolution. Chapter 20, illusions of fossil sequence. Chapter 21, fossil sequence and message theory. Chapter 22, biogeography. Chapter 23, cosmological issues. And finally, chapter 24 and 25, discontinuity systematics. In systematics and the origins debate. Now this, of course, 26 is conclusions. A lot of people may not realize, we keep hearing the term Baraminology in young earth creationism. The term came in part by Walter ReMine. He is the co-founder of the entire field of Baraminology. And to this day, I don't know that he's a professing young earth creationist. He said, he's sympathetic, but he felt his message theory had an independent life of the young earth creation debate. And I accept that.
Mm-hmm.
So he and Kurt Wise founded the entire discipline. And so he's been virtually forgotten, but some elements. So even though his name and his book has been, it's hardly ever talked about. And so I'm very grateful that you're bringing this topic to the forefront today. He is the co-founder of Baraminology, and he's also the co-founder of a lot of the population genetic research that we now know as genetic entropy.
Yes, he mentions that in his lecture on YouTube. He talks about genetic breakdown.
Goodness, see, so many elements of modern day creationism, as far as biology, it has its roots in Biotic Message, and then also this criticism of natural selection. That is pretty devastating because it anticipated some things that were happening. Not so much that they may have drawn on specifically Walter's book, but when Behe said Darwin devolves and showed that natural selection could actually destroy function, this was kind of the beginning of that. I began to see natural selection doesn't work that way, the way that evolutionists have said. So I'm really grateful. I was excited you wanted to talk about this because he's a dear friend. And when I read through this, I began to get excited. And then I actually had to stop and say, you know, I need to go back to school and learn this. So some of the stuff with gradual integrations, phylogeny, and where he covers the population genetics, I actually had to go to school, and it would be over a decade later, maybe a decade and a half, that I actually began to learn things like Fisher's theorem and neutral theory. And then I still don't know the cost of substitution, but I have talked to people about this. And the cost of substitution also relates a little bit to problems in genetic entropy in different ways. It's a related argument. It's not the same. So
Would that be related to the fact that natural selection has to select from?
I think the thing is like there are two costs. The cost of maintenance, meaning if you have too many mutations, you have to kill off a lot of kids to be able to maintain the population.
Oh, okay, so this is what Standing talks about a lot.
Okay, that's mutational load. There's a similar issue with the cost of substitution. So let's say you have a new mutation that arises. How are you going to get it to be fixed? Fixed meaning, you know, evolutionists just use terrible, confusing words. How do you get it to be present in all the members? They use the word fixed.
Yep.
So that let's say there's a trait like a new kind of lung or a new kind of heart. How does that spread through the population? Well, you have to kill all the rest of the population that doesn't have that trait.
Basically,
that's not very easy to do.
That's like the peppered moth scenario, where you have white moths when there was no environmental pollution. And then when the industrial age came, you could see all the white moths, but now you couldn't see the black versions. And so the white moths died off because they were picked off by bats or birds and stuff, and they were replaced by the black moths.
Correct. So the thing is that if there are all these single nucleotide changes that have to go through the population one at a time, and you have, say, 1 million of these, it's going to be very costly to the population. It may not be able to affect this change. And it's been explored some. And again, the more that I've studied population genetics, I said the more garbage that I keep digging up, and I just kind of gave up. But Walter was the first to alert me to it. So the first one, the one that I have highlighted on my channel, was the fiasco of Fisher's fundamental theorem of natural selection, which even Richard Dawkins was boasting that this is biology's central theorem. And most biologists won't be able to quote the central theorem, much less understand what it means. And it doesn't matter because it's not central and it's not a good theorem. But it gave the mystique that they actually have a real theory. And it's terrible. And it wasn't me picking it apart on my channel. I was just the messenger. I collected all the stuff that's already out there that's been critical of it. And now John Sanford and Bill Basner came along and they kind of piled on. But even at that point, a lot of population geneticists who are in the secular world could see its flaws. And they quietly just, they didn't raise the issue. They were honest enough to admit its problems. I credit them for that. But it would be published in journals that no one would see because they're in population genetics journals. And it's a very specialized field. And the popular science writers would not be jumping all over this. And, they don't want to say, hey, look how badly the evolutionary biologist screwed up again. They just don't say that,
Yeah. Mm-hmm. And it's interesting how a lot of comments from evolutionists on the Biotic Message said that this is one of the best summaries of the theory in print at the time, because he ends up digging up all this stuff and sort of gives a balanced view of what's being held to.
And I want to say something. I know this may sound kind of mean, but Walter was hoping it would get an even bigger reception, and I think I'm free to say this. I don't think he would mind. He was kind of disappointed that Kent Hovind was getting all the attention, and yet this book that had so much depth to it was virtually ignored outside of academic circles. The popular culture just was not enamored with this work as much as they were with Kent Hovind, the popular Christian culture. And
Yeah, it's not really, it has to be pared down. It has to be made granular and simplified so that you can take on these concepts at a pace that you're able to, master each one and then build on that. Because if everything's sort of locked in complexity and the depth, like you say.
And so let me just see if I could show something. There is just to let the reader know how packed this is. Let me see if they have a PDF. Yeah. There's this public PDF, and I'm going to share my screen. If one goes to the Blythe Institute, and I'll put this link in the video description, but it is my paper. It's just an educational paper. It's not. It wasn't intended to be anything groundbreaking. But I collected a lot of articles in the secular world that were critical of Fisher's fundamental theorem, and I go through the math, and I want to say, okay, that was maybe 2 pages in Walter ReMine's book, and I spent months learning this.
Yeah.
And perhaps that's why the book had some difficulty getting traction. It was so condensed.
Mm-hmm.
The density of concepts was just so packed that it's almost every 3 pages would probably take a year of study to gain mastery over. But that's also one reason I thought this was a tremendous book.
Mm-hmm.
Because you could just sense it was touching on the right topics. It was asking the right questions and going down the right paths. And I would get sometimes on the internet forums and start talking about this and the kind of the rude dismissals. I said, this is very disturbing because in mathematics, I never got rude dismissals like that. You just get you'd ask a question and someone would answer it straightforwardly.
Yeah.
Same with physics. But I would raise these questions, and there were just all these ad hominems and just trying to belittle me and use confusing terms. And I realized that's part of the deception, is to not be very clear.
Mm-hmm.
And just equivocations. I said, and then there was subtle circular reasoning, and I was like, oh my goodness, if we took every possible logical fallacy, not every possible one, but if we took like some of the most used logical fallacies and evaluated evolutionary theory on them, it's like most of it's built on that.
Yeah. It relies upon those fallacies.
So
Yeah.
So what you're saying is that sort of takedown where one sees that it's sort of a self-refuting theory when it's examined on its own terms, would you call that a deduction?
Deductive, yeah. That would be a deductive approach where you're examining the logic rather than a detailed review of evidence.
Thank
material
you.
evidence.
Thank you.
Now,
Yes.
Danny might refine that for us.
Yeah.
That's my just, because deduction is much more efficient when you're discussing things than the evidential requirement.
And that's what happened when I looked at this Fisher's fundamental theorem. I said it's vacuous and it's wrong. And of course, it took maybe 80 years before people were admitting it, and people were saying it, finally saying, "Oh yeah, it's wrong." I quoted Felsenstein in my paper. where he said Fisher's fundamental theorem is, and it's right here. It says, "Alas, not so fundamental."
Mm-hmm.
And
Well, it's interesting how these historical milestones form legs in the evolutionary table or chair. It's just like the Miller experiments. For years, everyone just quoted the Miller experiments without really understanding them and their veracity.
and they still do. David Neff in a discussion debate with me was quoting it. My jaw dropped. I said, David, this is this doesn't support your hypothesis.
Yeah.
So wow. Well, I'm really glad we had a chance to talk about this. And I think the viewers can sense it's like, okay, there's no way we're going to dive into this at all
No,
in
we
this one
probably have
show
to.
because it is so deep. Like I said, what was it? There was that one sentence on Fisher's fundamental theorem. Yeah, right here. Some of the issues such as Fisher's theorem. Okay, so that was 3 months of work for me professionally.
Mm-hmm. Well, it's nice to see you do that hard lifting, and maybe. But the thing is, to then take that and simplify it and present it in a consumable
Yes.
way
Oh,
is another level of difficulty.
well, one of the problems is the obscurity and obfuscation is part of the illusion. Meaning
Yes.
they throw so much mathematical theatrics. He said no one would actually do this seriously unless they really believed it. And I'm just like, well, maybe they believe it, but they're half delusional too, because
Yes.
the math is irrelevant. It doesn't describe reality. Can you give me I'm going to pause for just a second. We had to take a commercial break. So I have no clue where we left off here. We were talking about Biotic Message and how dense it was. I don't
The
know.
theorem, the fundamental theorem. I think we were talking.
And so Walter's work is very, it touches on so many things and it only delves into it in a few pages. And then, but it still outlines all the various ways, almost any of the ways that evolution could be taken down that he outlines would be almost sufficient in and of itself to knock it out.
Yeah.
And I have pursued the molecular level stuff. Walter laid a little bit of the groundwork for the genetic entropy argument. That's powerful in and of itself. And then he laid out the origin of life argument, saying that other people are going to take up the banner, take up the cause, and we're going to own that argument. We already did, and we own it even better now. So some of it was prophetic, and some of it is ongoing. So
Yeah, the book really represents a master point of view
yeah,
on the theory
yeah,
that's really unequaled in a lot of ways.
yes.
And if you watch, have you seen his YouTube presentations?
No, actually, I saw part of it, and I'm sorry to say this, Walter, if you're listening. I actually was too bored at that point to keep moving on, and I didn't understand it. And this is before I met him. And so it's like the theory grows on you the more you're exposed to it. You begin to appreciate it in ways you didn't before. And
So how did you actually meet Walter?
we met through email lists at Access Research Network, which is an old, it was a forum, an electronic forum in 2003, 2004, 2005 timeframe.
I've seen that. They have some wonderful videos.
Yes. And they had a good debate forum, and Walter appeared a little bit and was abused by the evolutionists there, but I got to meet him, and he talked about Haldane's dilemma, and that was the beginning of it. And we started to email. And then on one of my casino trips all the way up to St. Paul, Minnesota, I said, hey, Walter, I'm going to be in the area. How about we get together? And we went out to a restaurant called Leanne Chin. I still remember it. And that first night we met, we talked for 8 hours.
Wow.
Going in and out of the restaurant. We just didn't have enough to talk about. And and then so I went on a couple more gambling trips up there. It's one of my favorite casinos. Little Six in Mystic Lake. Oh my goodness, I love Mystic Lake. um, and I'd meet Walter whenever I was on the casino trip. And so we had always these long conversations that would go very detailed. And so that's how we began to talk. I regret to say that because I've been basically I was taking care of mom pretty much for 17 years, but I had help. But then the last 7 years, it's been only me. The last 8 years, 7 or 8 years, and so I haven't been able to travel. I've been working from home, and thank God for Dr. Sanford that he provided me an income where I could work from home and do research. So that's kind of the sad story that I haven't been able to stay in touch with Walter. He briefly came out to a conference on the East Coast, and we got to talk for just a little bit. I still remember he and I and Rob Carter were just talking about Y chromosomal atom and this thing about mitochondrial Eve, and I learned a few things. But sadly, I missed. I had to go home and take care of mom, and Walter had a magic show that I missed, and I'm really disappointed I didn't see it.
Wow.
So when you mentioned Walter using magic, and then he began to see evolutionary theory as basically illusions, I said that was interesting. That's something I didn't know in all the time that I've spent with him. Now, Walter's ideas he's been critical of mainstream intelligent design theory, and he's been critical of creationists. So he's been kind of a little bit out on his own. And but there are people that are very appreciative of all that he's done. And and you being one of them, I'm just very grateful. I'm thrilled we're talking about this. Sadly, when he has, when I'm just looking through the book here, I was like, oh my goodness, some of this I don't hardly touch. So let me share my screen again. So like chapter 3 has been the focus. Chapter 3 here has been a big focus for just like 2 years. And that was with some top rate, Ivy League level, Ivy League trained scientists basically doing chapter 3, and that would be Change Tan and Rob Stadler. So just chapter 3 alone has just borne out. But it would be interesting to see what he had to say there. The chapters 4, 5, and 6, 7, 8, and 9, I kind of lumped them together. The whole study of population genetics, the idea of fitness, that's still being worked out. Now we've had a victory here on Fisher's theorem, because at the time that still had a lot of credibility, and about 25 years later, finally the mainstream is saying, oh yeah, it's not so fundamental and it's not very good. And I'm like, yeah, okay. So it's taken 80 years, 100 years to finally self-correct. And
so how was it actually, what was it actually producing, that theory? How were they utilizing it? Or were they just referencing it as a proof of some sort?
First off, it was used as an illusion that they actually solved anything. So it's like, oh, the gravity of a mathematical theory that says there's a fundamental theorem of natural selection where everything improves. And it's like, okay, yes, it does in this idealized environment. And then also you redefine what improvement means. So there's a sleight of hand. If you look at the theory,
sleight of definition.
Oh my goodness, it was terrible. So let me give a concrete example. We know that we have this phenomenon of pangenomes, which is also troubling for evolutionary theory. It's especially bad for multicellular eukaryotes, but we have a model of it in bacteria where you can take 80% of the E. coli genes that are, and just dispose of them, and the E. coli will live. And it turns out a lot of E. coli strains have just dumped their genes. So natural selection favors losing genes. Now, thankfully for the E. coli, if you could say that for a bacteria, thankfully, for the E. coli species, they have because they're bacteria, they can have horizontal gene transfer. They can reacquire some of the genes that they have gotten rid of. But you could see that this would be devastating for a multicellular eukaryotic organism that doesn't have the ability to as easily acquire genes from other creatures. I don't know what the status of that is, but it could be so hard that it virtually doesn't happen. So how does that relate to Fisher's theorem? Well, in Fisher's theorem, the most reproductively successful will keep reproducing and be successful. It doesn't matter if it's lost its genes. And so we have examples of particularly what we call symbionts or parasites. They'll join the host. They'll start losing organs and genes. They'll never recover them in that line. So if they're not bacteria and if they don't have some horizontal gene transfer mechanism, which I think in certain cases it's very doubtful. So Fisher's theorem will say, oh, that's an improvement, it's inevitable, but yet it's totally disconnected from reality because it's a sleight of hand. That was the point of Darwin Devals by Behe. He was saying 90% of the beneficial mutations are loss of function. Loss of function. And so I tried to put that in slightly more concrete terms. So let's say that you have one gain of function, like say you improved an IQ point. What good does it do you to have that when you have, like, say, a decline in eyesight, loss in kidney function, diabetes, a little bit more tendency for diabetes, overweight? And you can list, so just say hypothetically for every gain, you list 9 things that are wrong that you lose. How can you have evolutionary advancement in a situation like that, where let's say all these 10 genetic losses are also counted as quote unquote beneficial? That's going to be really bad, because then natural selection will be favoring the destruction of your body slowly. And we have very good examples of this, very well established experimental and observational examples. The only place the opposite happens is in the imagination of evolutionary biologists for the distant past. Nowhere do we see any example of it in direct observation in the lab or the field. And what we see in the lab and the field also agrees with theory. Michael Lynch said it well, and I have to wonder if he wasn't reading ID literature. He said, it's so much easier to break a function, and there's so many ways to break a function than make a function. And it's like, yeah, that's a really good summary of exactly the problem. And so Fisher's theorem will actually demonstrate you're going to have genetic loss, but they don't represent it as that.
So what would be the prevailing reason for the tendency towards simplicity with selection?
Complexity is very hard to arrive at when you need many simultaneous changes. So if the change, they call this, let me see if I could find what they call a fitness landscape. So I'm gonna.
So he talks about that in the book, The Fitness Landscape.
Oh, see, the guy is just on the ball everywhere.
He talks about you can go up a plane, but then you may have to come down a plane if you are basically fixating something that's unbeneficial, or there's no progression beyond that point.
Okay, so I'm going to show an example. Conceptually, this is a fitness landscape. Here's a very simple fitness landscape where the idea is when you go up, there's evolutionary pressure pushing you up. It's kind of like anti-gravity, okay? Just the way that they make the diagram. It's like, okay, yeah. So Fisher's theorem, et cetera, would say, oh, wherever you start here, you're going to end up at the top of the mountain. You maximize. So to answer your question, why is there this tendency towards simplicity?
It's an accurate representation of that, because the base of the mountain is wider than the top.
Yeah. Where
But
it gets,
to me, it makes sense in the sense that, you know, if you have simpler system, it takes less energy to maintain. That would be one of the biggest going concerns, is energy usage.
yeah, and let me try to think about this again. I may, apologies to you and the reader, I may have been going down a rabbit trail trying to answer your question. There is one aspect of this, if we just talk about complexity. So let's say one is at this little peak here, and so we have a creature that's doesn't have any of these capabilities, like say electric sensing. Say it doesn't have electric sensing, or let's say it has a two-chambered heart, and it needs to evolve to a four-chambered heart. Well, if you start redoing the plumbing, you end up losing fitness, meaning to get all the way up here where there's more complexity, you have to sort of destroy the existing system, and that's going to be actually selected against. So what'll happen, okay, maybe this is a better way to frame it. Natural selection would actually prevent the evolution of complexity. So that's the first aspect. Natural selection prevents the evolution of complexity. Stephen Gould, or someone even earlier than him, said, what good is half a wing? So something that is an arm will probably be a bad arm before it ends up being a good wing. Or we could say that for a lot of organs, that half formed is actually a liability. And we definitely, you know, in the morphological world, evolutionists might get away with it, but I definitely see it in the molecular world. A protein that, like say topoisomerase, if it tries to evolve to another protein, it's going to kill the organism.
Yeah.
And ironically, Dr. Dan and other people have been saying, yeah, we don't accept the idea of universal common ancestry of proteins. I said, it's understandable, because there can't be macroevolution from one major protein family to another, because otherwise the protein, especially if it's like critical, would lead to the death of the organism. I could clearly see that at the molecular level. So what does that mean? Natural selection will prevent the evolution of greater complexity. The only route available to it is if reproductive fitness is tied to loss of function. Maybe I have to think about this more, and apologies to you and the reader, that your question is a good question, and I have to, I'm sure we can formulate a way of stating it more than the way I'm fumbling through it now.
Well,
but the important thing is, yeah.
In order for natural selection to progress with complexity, each stage that it's selecting for has to be beneficial.
Yeah.
But if you're trying to put something complex together, it has no beneficial function until it's actually together and completed.
Yeah, and I think I can show maybe, you know, a lot of people quote Doug Axe's work, and God bless him. I'm not trying to be too critical, because his work is 20 years old. There's an updated view of this, and I gave a presentation recently, and I'm working on a paper, and I don't mind sharing it because I've talked about it, and it's basically, it's really just common sense. So let me see if I can find it. I'm going to show some pictures here. And if you've seen my protein probability talks, yeah. So I'll show the screens that aren't too sensitive. So a lot of people, a lot of ID people in the popular, kind of in the popular realm, are using Doug Axe's figures. And what I've tried to say is that was good for the single protein family that he studied, but there are way more other proteins than what he studied. And the problem becomes really obvious when you actually look at other proteins. So they keep citing this figure of 1 chance in 10 to the 77th power of a protein being formed by chance. That's specifically a beta lactamase. But we can look at, when we look at other proteins, the problem becomes brutally obvious. And let me share my screen here. So a way to visualize the problem of proteins is just looking at this toy. This is an interlocking toy. I guess it's to train like preschoolers or, you know, some sort of young kid in being able to assemble things, and it's kind of like a jigsaw puzzle, but in 3D. And you can see how all the parts have to fit just right. And it turns out that's the case for what we really mean by functional proteins, because functional proteins, to be functional, have to be integrated into a complex. So here is a picture from Bruce Albert's Essential Cell Biology, figure 2.14, 5th edition. And it says a large molecule such as a protein can bind to another protein through non-covalent interactions on the surface of each molecule. And I'll skip. Shown here is a set of electrostatic attractions between complementary positive and negative charges. So first of all, they have to be shaped properly, and then they have to have charges. The amino acids have to be presenting a subtle charge to allow this kind of bonding. And there are other kinds of bondings than these kind of ionic ones. And so the idea here is kind of similar to this. Now, this is just kind of a random conceptual drawing. I'm going to show some actual proteins. This is a homohexameric helicase. Hexameric means it's made of 6 parts, hexa meaning 6. And so each of these parts here, see like the blue, the green, the purple, the blue, the green, the purple, each of those separate colors is an identical copy of another one that's in a different color, identical. So there's 6 pieces, and they have to connect. Now, it's not obvious that these are very intricate connections here, these connection points. You could kind of subtly see that there's that shape there, and people who are structural biologists would really know this. These are non-trivial connections. The connections have to have a precision on the order of angstroms, which we can't even achieve in any machine that we build. It has to be that precise, the distances. And so you could see this, it's kind of a subtle hexagon. And the reason is, even in the man-made world, it makes sense to have hexagonal structures. The symmetry is very important. And then here's a topoisomerase. And I actually examined the number of connection points here. It's about on the order of 50. 50 amino acids have to be perfectly lined up in each. So the red is an identical copy of the blue. And it's like, okay, so that's what functional means. It has to actually, things have to physically, mechanically fit. And then not to mention, it has to be, you know, a designer would have to conceive of like basically how the scissors would be put together and how it would function as a whole. And so this parallels a lot of things in engineering. So that's why I say you don't have to cite Doug Axe's work anymore. You can actually visually see what the complication is in making a functional protein. Because if you knock out enough of these, this thing's not going to function. Not to mention, it's not a matter of just making a shape. It has to be a shape that is connecting together and actually operates. So this topoisomerase here will actually does literally act like a scissor. It untangles the DNA by cutting it and then reconnecting it. It's phenomenal. And then there's this potassium ion channel. It's made of 4 identical parts. It has to be connected again with angstrom and sub-angstrom precision. And you could almost see here the outline of a square, and you could see that that's a square nut with a hole in it, and you could see the symmetry there. Now, going back to the little toy model, I'll show one of how a protein folds to itself. This is the EZH complex. Do you see how everything kind of has to fold and connect to each other with the colors here?
Mm-hmm.
This is non-trivial. So when this protein is expressed, it has all these domains, and when it folds, it's going to fold all these pieces beautifully together. And you could see here the little toy model kind of echoes what you see at the protein level. So with that understanding, it's very difficult. Okay, so let's say we were going to try to mutate. They try to say, oh, it's so easy to mutate the protein. It's like, maybe you'll find 1 or 2 that it's really easy to mutate and get new function. But what's going to happen here? If you start changing this, you're going to have to change this and this and this to get it to all fit together. And that's definitely going to be the case with other proteins like this. If you change it, it's going to change the connection points. It'll change the size of the hole there, which is really critical. It's going to fail if this hole is just a little wider or a little smaller. It's going to fail. The size of that hole is really important to selecting potassium ions. And then same for here. And so I'm just trying to show visually.
How far up is this above the molecular level?
That is,
like, do each one of those proteins have like an atomic weight based on
okay,
a specific structure?
Let me forward through it first, because I don't want to show anything too sensitive. Okay, so far these are things. Okay, I don't want to show that. Okay, so I showed the topoisomerase. Let me see if I could show the topoisomerase. Okay, I'm going to show this screen. So your question is, this is the protein here. A nice term is to say, call it a polypeptide, but we can call it a protein, because sometimes it's ambiguous whether we're referring to the protein complex, because we can also say these proteins form another protein. And one way we can distinguish it is say this polypeptide, the two polypeptides form a functional protein complex, and this is the topoisomerase. So to answer your question, the individual amino acids can be spelled out here. I don't know if you could see my highlighted cursor here.
Yep.
So MEVS. So M is methionine, E is, I forgot what that is, and I'm embarrassed. V is valine, S is serine, P is proline, L is leucine, Q is, I think it's asparagine. I have to go back and relearn it. It's only an alphabet of like 20, 22. But so you could see that this one has about 1,500 amino acids. And so this would be a molecule of 1,500 amino acids. Does that sort of answer your question about?
And the amino acid, how high above the molecular level is that?
An amino acid is a molecule. And let me just answer. This is really good. I can't tell you how thrilled I am you're asking, because I wish creationists would express this level of interest to understand these details. So,
yeah, I usually get into unlocking truth, and it all starts with a question. You know, you unlock.
Yeah, I'm thrilled. Okay, this is from Leniger Principles. I had an old school professor, God bless him. When I took biochemistry, he said, I want you to recapitulate these diagrams on your exam. So I had to, it actually took me 15 hours of memorizing these amino acids. So let me go back to my, okay, so let's look at methionine, the M, methionine, and then we'll look at the, so methionine's right here. So you have let me see if I could zoom in. You have so this is methionine here, and you have C, which is carbon, O is oxygen, H is hydrogen, and you could basically see how it all connects. So this is at the atomic level, and then so that one molecule that I was showing, it's summarized by M, and then we have the E. I'll just do one more because it's going to be very tedious to go through this. The E, oh, I don't have the name of that. Hang on. This is bugging me because I should know this since I'm supposedly a protein researcher. FASTA format.
You don't give them all equal attention.
No, I focus on the serines and threonines, and I'm going to find out what that E means, because I just can't let that go, especially we have viewers that we're trying to educate on science. So an E, I'm looking at my other screen here. E is glutamic acid.
Oh,
And
really?
Q is glutamine. So glutamic acid is an E. So I think it's this one, glutamic acid. It can be also glutamate. And so you could
And
see
it has glutamine as well,
glutamine is
or glutamine.
Q. Yeah. So I think glutamate is glutamic acid. There's a subtle difference between it being glutamate and glutamic acid, but it's basically, we could say it's the same amino acid because it corresponds to the same codon. I'm pretty sure that's it. If someone who actually knows this, and I misstated it, feel free to correct me in the comment section. But the point where we were headed with all this was, so you have the M, E. So you could see each of these letters here is several atoms.
Mm-hmm. And these are the pieces in the toy that have to fit together.
Yes. And so, yes, randomly we could make something that's just garbage and it's just a string. But
these aren't racemized, are they? They're all chiral.
Yes, they're all homochiral. In fact, you won't, what this diagram here, it's called a Richardson ribbon diagram. Anytime you see these, like, it looks like a ribbon, that's an alpha helice. And if you didn't have it homochiral, you wouldn't have that ribbon. And that ribbon is really important. Your protein would basically be useless without it.
So what is the bonding point on homochirality? It's not ionic, is it? Because that would be
it's homochirality is referring to the amino acid itself. And Dr. James Carter, Dr. James could better explain that it's the configuration. And I'm really glad you're asking this because, oops, I don't want to show that. Let me just kill that because I don't want to be showing there are a couple slides that were a little sensitive there. So chirality, amino acid. So one could see how this, Walter just mentioned origin of life and chirality relates to it. And so this is, let me see if I could shrink this down. So the actual configuration of the molecule will be different in terms of where everything is in 3 dimensions. Mm-hmm. So even if it has the same chemical formula, it's going to have a different 3-dimensional structure because of the way it's connected up.
So Rod Stadler, he was saying that some of these things have like 16 different chiralities.
That's for nucleotides. This is an amino acid. And yes, that's a very severe problem. That's one of the best parts of the whole book that was analyzed.
So is that a more complex structure that's higher up the scale?
It's more primitive, but it's more complex in the sense that it's more fragile to assemble it. I wouldn't say more or less complex. It's more improbable, far more improbable, because if you have like all these different configurations, in fact, it was just between adenine and cytosine. Yeah, adenine and cytosine. There's 16,000 ways to connect it. Whereas,
and only one works?
Yeah, only 1 or 2 works.
Okay.
Yep. It's a severe problem. Well, hypothetically, we might create life with a different organization, but you still have to have it all lined up. And I covered that in one of my talks because it makes it readable. It makes it machine readable by all the machines that do DNA replication and transcription, and also repair for that matter. So where were we? We were talking about protein complexity, and I tried to show visually why it's difficult. You can't, these evolutionists will just cite one measly experiment where they happen to change function, but most of the time it's just going to break it for the reason like, okay, if I just took let's say I just took a hammer or a chisel and I randomly took car parts and just started hammering away, uh, it's not going to fit. You're going to break the function. It's so much easier it's very hard to change a spark plug that's very well optimized for its function to make it do something else. And what they do is they cherry pick these trivial examples where they achieve new function. They never show all the other issues where you totally just destroy the function of the protein. And that's very misleading.
So they're exploring, well, you have, like you said, thousands and thousands of possible ways of bonding things, and they're going through 2 or 3 of the successful ways of breaking it apart and recombining it, when there's thousands of other ways that are, detrimental.
Absolutely. And in my debate with Dr. Dan, he was citing 3 beneficial mutations. 2 of them were loss of organs or functions. One of them was the elephant tusks being lost, and the other was sickle cell anemia. So I said, well, that's not really a good batting average for showing beneficial mutations. And I was laughing. Perhaps in another video, I said, yeah, it's so hard to get a true beneficial in the sense that it adds to reproductive success and it actually adds a significant addition of complexity. It's so rare. And you could see
the interesting thing about that is that that's so high up the scale of things, and in order for there to be any change, you have to have a reconfiguration at the molecular level.
Yes. So let's just say, okay, say you're going to try to build, say hypothetically, topoisomerase didn't exist, which is kind of ridiculous because for a lot of life, the creature would not even be alive if it didn't have topoisomerase. So let's say helicase. How many ways? One thing that was probably easy to see is that how can you mutate something so that it can rearrange itself and reconnect to itself?
Well, how can you have variation within chirality, chiral structures? I mean, if the only one works, then how can you change anything and still maintain function?
You can't. Now, chirality, evolutionists will say, well, that was, we'll assume abiogenesis and that chirality problem is solved. So once you have a genetic translation system and you have a basic metabolism, everything will be homochiral. And it's like, okay, let's just grant that for the sake of argument. But then how do you start building multimeric proteins? These proteins that need several simultaneous parts to exist for the protein complex to work. It's just like systems in a car. A starter, like say the starter of the car, it makes no sense if you don't have battery cables or a battery or an ignition system. And then the starter has, the analogy to mechanical systems and engineering is you run into the same problems times 10 at the biological level. And that's one reason evolutionists don't like hearing the cell being described as a machine. So there's a big argument on the internet. One of my detractors is just trying to assail the idea of cell as a machine. And I just laugh because one of the top 20 medical researchers, let me see, Michael Sheets, cell. Yeah, the cell is a machine. So let me show it on. Let me share my screen here. He has this book, and he's one of the top 20 medical researchers. He wrote a book, The Cell as a Machine. And it's really funny. He has a picture of a car there.
Yeah. Very telling.
Yes.
I'd like to know more and understand more about how chirality structures actually are connected together at the amino acid level, because I sort of understand how these things don't happen naturally. But is it because there's they're polar, their
Okay,
polarities?
so first off, there are two issues of chirality, one at the nucleotide and one at the amino acids. So DNA is made of nucleotides,
Hmm.
and DNA is, when it's translated, it be, it makes proteins. The proteins are made of amino acids. So we have to be careful here which particular class of molecules we're talking about. Are we talking about amino acids or are we talking about nucleotides? Each of them have severe problems. The more severe problems with nucleotides, that's not going to be solved. I don't anticipate, because if we could have solved it, we would have solved it by now, because we'd be able to do a lot more biotech, genetic engineering so much easier if we could solve that problem. And Change Tan's very sharp. She picked up on that. So maybe I have an older lecture and I could show you the importance of it. If you give me a moment, and then maybe
Sure.
we could just reconvene sometime and
Hmm.
talk, because I'm kind of running out of gas.
Yep, I understand. Yep.
Structural bio. I had a talk on structural biology. Let me find it. And I'm just really thrilled. So, by the way, I want to invite you to the Evidence Reasons Academy. It's not up and running yet, but where I teach basic chemistry, and
Hmm.
it's for free.
Yeah.
Part of the reason I'm doing this is so I could actually learn myself. So even though I may not even have any size of an audience, I'm doing it mostly for my benefit, because one way to learn is to actually teach. So I'm
Hmm.
creating these lectures partly for my own benefit. And if people end up watching it and also learning something, then all the better. So let me share something here. And I'm just going to conceptually show what the problem is, why it's desirable to have nucleotide chirality. I'm not talking about amino acid. The amino acid chirality is probably a little harder to explain. We'd have to go into some structural biology, and that's like a 3-hour lecture. In my biochemistry class, that was a 3-hour lecture on the importance of homochirality and amino acids. But it's a little easier to talk about it in terms of nucleic acids for DNA. So it says here, you could see here, we could read the letters very easily because it's nicely organized.
Hmm.
Where it's not easy to read the letters is if you have a pile like this, it's not readable. You have Scrabble letters that are flipped down, they're in the wrong orientation. So when Rob Stadler was talking about all the ways it could combine, this is what will happen when you have the nucleotides just kind of randomly combining. It's not a nice linear molecule. And so I had a picture here of Heath Ledger or
Oh,
accounting ledger.
one point you should mention is, why are they combining? What is the nature of the combinations? I mean, how do they connect to each other? Or is it a ionic connection?
Covalent bonds, and they naturally form. In fact, when we're trying to build DNA synthetically, like Craig Ventnor did from a computer program. So he just said, okay, this is the DNA sequence I want. He goes to a laboratory, and the laboratory has to do this. They actually have to prevent certain covalent bonds from happening, and that's in Dr. Tan and Stadler's book that explain the process, how they have to, because if we just threw all this in a soup, it's going to combine spontaneously in a way you don't want. So just imagine you have all these Scrabble letters and they have a little bit of glue on them, and you just dump them together in
Hmm.
a stew. It's going to connect in a way you don't want. So what they have to do is kind of, it's a very long, painful process where they'll take one class of molecule and say, okay, we're going to connect it to our pre-existing strand. We're going to make sure it's oriented the right way, and then we're going to glue it there. And then we stop the experiment, clean out the quote unquote beaker. It's really an assay. And then like the next letter, we put in all those chemicals and we do this process. It's very painful. It's extremely slow. It's not very reliable. We can get maybe 100 nucleotides through this process. And then when they get 100 nucleotides, they have to stitch them together. It's just a nasty process. And she goes into brutal detail in her book how this happens. But So suffice to say, just imagine that you had these Scrabble letters and they had a little bit of glue on them. Now just throw them in a box and shake it. You're not going to, it's not going to be nice and organized like this. We want to have it in a nice linear molecule. This is not exactly linear, but you could see it's easier to read. You want to have it in machine readable format. And what I mean by machine readable format, see, everything's nicely organized and uniform. The molecular machines that do transcription, replication of DNA can move along this, and I can, we can show some videos to that effect. Hopefully I could find some public domain ones. But again, just think of the word ledger. There's Heath Ledger. And we're just going to have a Scrabble letter that has it. It's nice when it's a nice linear arrangement like this. But let's say the Scrabble letters were even slightly disoriented or misoriented. It's very hard to read it. In fact, you can't even read this one because it's flipped the wrong side. That's analogous to the problem of what happens if you don't have, what they didn't call it, not just homochirality, it's homolinkage. So apologies to the reader. I should have said homochirality and homolinkage, because what's desirable is everything is lined up properly. You see, there's a natural orientation. If we just threw these Scrabble letters on the floor, it's going to look more like that. When if you wanted to have it, you want it to be readable, everything has to be nicely arranged so it looks like that. And it's kind of the same thing at the machine levels. So that's why not just homochirality, but homolinkage for nucleotides.
So the chirality doesn't apply to each letter, does it? Or does it?
Yes, it does.
So yes. there's an individual chirality, and then there's a bonding chirality.
Bonding linkage. So they used a term that you won't see anywhere in literature because they don't even recognize it as a problem. It's called homolinkage. So let's say you had something
that was a covalent bond.
Yes. So let's say you had something that was linked improperly, and I'm just going to illustrate that here. Yeah, this would be very bad, where you link the wrong side. You link one nucleotide in the wrong orientation. That'd just be a disaster. It couldn't be readable at that point. And we know that from physics and chemistry, that it's just going to jam.
So you bring in the idea of reading, which is very interesting. We get to the sort of the immaterial idea of information.
Yeah.
So the bondings, the links have to be in a way so that they're readable, so that they
exactly. You get it. What you just said is exactly one of the problems. And the thing is Dr. Tan and Stadler have identified so many problems. It's like any one of these is enough to sink the origin of life problem if you assume naturalism only. Any one of these would break it. And
so they go into detail on the covalent,
the
covalent bond?
Yeah, and that's why I'm actually trying to study chemistry again, so I could actually understand Dr. Tan's argument more thoroughly, because she actually okay, so first I have to learn things like this. This is a nucleotide here. And she says these are the various nucleotides and how they bond together. So you could see here the covalent bonds, and they call them phosphodiester bonds. Those are covalent bonds that bond one strand, and then the complementary strand is here. And then over here are hydrogen bonds between the two. This is really complicated. I mean, this is like one should take organic chemistry, and then one could understand this, and then biochemistry. So this is like 4 semesters of chemistry to be able to appreciate how this is put together.
So what would be the simple definition of a covalent bond?
I don't have one off the top of my head.
Oh.
And that's why
that's what has to really take place. There has to be some real clear, since that's where the problem lies, you really have to understand that concept very clearly.
It helps, but what Dr. Tan was illustrating, she was saying, what happens if you had the wrong covalent bond? This is like, again, I tried to illustrate it by analogy with the Scrabble letters. Let's say the Scrabble letters in the wrong orientation, and then you glue it together. It's not readable. So maybe that's a good way of explaining it, is that if the covalent bond ends up being like, let's say we glued the Scrabble letters together, it's no longer readable if you had this. That's very easy to happen.
What is the nature of those linkages? I mean, is it an ionic thing?
Covalent. They're covalent between the nucleotides. So let's say
So what's the difference between a covalent bond and an ionic bond?
That's one reason I'm having my chemistry series. The
Oh,
covalent
okay.
bond has a lot higher bonding energy than an ionic bond. And technically every ionic bond is slightly a covalent bond. And that deals with quantum mechanics. But the main thing with the bond is there's an energy. It takes energy to pry it apart. It is, I wouldn't want to say glue, but it's a strong connection.
So it's something other than just polarity.
Yes. Yes. So this line here indicates a bond. So this nucleotide here, it has this particular molecule at what they call the 3' position, and it's going to connect to this
Phosphorus?
phosphate group, right?
Phosphate.
And then the phosphate group has to connect to the, I think it's either 4' or 5'. It looks like 4'. I didn't know that.
So these are nucleotides.
Yeah.
They have connection
let me see if
points
I
on
can. Okay, so the nucleotide, do you see how I'm circling it? That's a nucleotide there. And then this is another nucleotide here. The way you can identify the nucleotide, you have the nucleobase, which is colored. And then you have the standard deoxyribose here, and then the phosphate group here. And so that you put the three together. Let me see if I had the diagram. Oops. So the diagram is you have a phosphate group, the deoxy sugar, and then the nucleobase. And the bases could look like that. So
if we broke down the word covalent, valent is referring to the outer shell of the atom, isn't it?
I tell you what, you're asking such good questions. And I mean this sincerely. I'm not doing you a very good service the way I'm kind of just doing this off the cuff.
Yeah, I'm just your feedback loop here.
Yeah.
You're trying to explain something, and I'm giving you the feedback every step of the way.
Yeah. So a covalent bond is a chemical bond that involves the sharing of electron pairs between atoms. Now, this whole thing of sharing electron pairs, that goes into some really deep physics of Schrodinger's equation and quantum mechanics. As far as a chemist,
that's not opposites attracting. That's something else.
It is something else, and that's why, from the chemistry standpoint, we'll just say, okay, we know it takes energy to break it apart, and we can measure that energy, and it affects the position. The physicist will say, okay, what's the mechanism of this? And it deals with quantum mechanics, which is really deep. I mean, it's just brutally hard, and so there's some things where we'll just accept it, kind of like, okay, you throw hydrogen and oxygen together, and it'll make water when you throw a spark. And it's, for a chemist standpoint, it's like, okay, we know that it's creating covalent bonds. The nature in terms of the physics.
How does the chirality relate to the covalent bond? Like, is there racemized covalent bonds and non-racemized covalent bonds?
No, it's the configuration of the covalent bonds in chirality. So I don't want to show that screen here.
So you have the left-right hand pairs. Everything is all...
For amino acids, it's easier. I'm not very familiar with the chirality in nucleotides. So apologies in advance.
Sure.
So this is a famous diagram. It's just like everywhere. So the hydrogen has a covalent bond to the carbon. The carbon has a covalent bond to I think that's the carboxyl group, and it has a covalent bond to the amine group, and then this R group, which is unspecified. That's what makes the amino acid with the R. And so the covalent bonds, it's the location of the covalent bonds with the other partner. So you could see here that the H connects to a different part of the carbon from the left versus the right when you look at the configuration. So homochirality is the configuration of the other molecules and their covalent bonds.
Mhm.
Just think of it in terms of the geometry of the entire molecule that is covalently bonded with all the atoms there.
And are these naturally occurring, these left and right hand pairs? Or are these
It's naturally occurring for it to be left and right if they exist.
if
It's
they
not a
exist?
given that they're going to exist in nature for very long. And if they're left-handed, they'll spontaneously become right-handed. That's the process of racemization. The process is that these molecules are probably vibrating because of, well, to use a colloquial term, it's the heat energy. The proper term is internal energy. So as the temperature goes up, there's more vibration and collision. And as those collisions happen, the covalent bonds, the configuration of the covalent bonds has changed. I actually don't know the mechanism of how come these molecules would spontaneously just rearrange themselves.
Mhm.
That's an interesting question, how it is able to achieve this without losing energy. In fact, this is kind of obscure. The Gibbs free energy says that it's more natural for, if it's all left-handed, to become a mix, 50-50 mix of left and right-handed.
Mhm.
And there's some more subtleties to this, what they call not just racemization, but epimerization or something like that.
Mhm.
And so when you ask, does it happen in nature? What would happen in nature is spontaneous isomerization, that you would normally have a 50-50 mix. If you started out with all left-handed, they would eventually become 50-50, 50 left, 50 right. And
So the prebiotic or the abiogenesis is all about the natural creation of these compounds and them creating proto structures that eventually start resembling life in some way. But the idea of them naturally occurring uh, and with the right chirality is very low.
correct. And I tried to show this conceptually what the problem is. See if I could
Yeah.
find it another way. Because I did cover chirality, but there are other. Oh, look at that biotic message. I ended my talk on that, at least the slideshow with the biotic message. Let me look at my.
I can see I'm gonna have to get their book, The Spiral,
Yes,
or The Staircase to Life,
Stairway to
just so I
Life.
can
Oh,
start to
yes.
speak this stuff so I can understand it, because
Yes.
there's an idea of accurately representing information, but then there's the idea of conveying information in a way that is consumable.
Yes. And I want to say this. I believe that trying to solve all this in terms of 1 hour debates is just ridiculous, because you give the
Mm-hmm.
other side an easy pass. The, the strength of the design argument is not in anything of it being consumable. The strength is in the actual details. When the other side has the opportunity to kind of gloss over these important details, that's how they maintain the illusion that they're winning the argument. And that's why I said, some of these debates, these 2 or 3 hour debates, I said, that's ridiculous. You want to talk about the origin of life? How about we have a 50-hour discussion and
Mm-hmm.
we break it up over a year? Because when we go into these details, we basically go through the chapter by chapter discussion of the stairway to life. That's when the other side just blown away. It's like what you're asserting is ridiculous. So the reason I have this picture up here of the cards, you see the tent cards.
Mm-hmm.
This is conceptually, kind of metaphorically saying this is the problem of design. Random orientations and starting points would not make anything like this. See, if we
It's too
just took cards
fragile.
and randomly put them down, they'd lay down like that. It's very unnatural that they would be like this.
Hmm.
And that's metaphorically what the real problem is for the origin of life. It has a very special kind of organization that puts it in this very delicate state. That's why I love this picture. I love pictures like this because it explains what the real problem is that it's possible these molecules can be put together this way. There's no question we wouldn't be alive, but why should it arise naturally in a random prebiotic soup? This would be like expecting the cards to spontaneously assemble like this.
Yeah, you'd have to understand what all the factors in the environment that contribute to any sort of configuration that the
Right.
cards might have, and you'd have to say, now what formula or configuration of environmental factors is going to bring about a likelihood like this. And once you start going through them all, you find out there's actually none. There's nothing that would. There's no factors I could configure to do that.
Yes, and that is the principle of the biotic message. So if I wanted to convey design, okay, we know that the cards are man-made. That's not. But if I wanted to say, I really wanted people to know that me as a designer, I want them to know that this wasn't accidental. You could do that. You would just say, okay, I could build any number of structures, probably infinite number of structures. Just, one could be very creative like this, and it would convey to the observer that this is not an accident of random accidents,
Mm-hmm.
that this isn't a product of random accidents. And what Walter ReMine said is biology specifically was constructed to convey the message that this is designed, that no natural process could assemble it. And whatever, it's designed to resist any attempt at trying to explain it in terms of natural processes being just random chemical actions that we
Mm-hmm.
could describe in terms of distributions of how chemicals behave. And that's the takeaway from the biotic message. Every time I was in a debate with an evolutionist, and at the time when I was debating this, before I got involved, before I even had a advanced physics and biology degree, I said I could always count on the biotic message, because when they would propose a mechanism, I always had a feeling that God designed it in a way that would prevent evolution from being an explanation. I could always count on that. I said I just have to look long enough and hard enough, or someone has to come along and provide the data. And I was up against these professional biologists and people that have been defending evolution for decades, and they would always fail. And I said, yeah, God really did make this very hard for them to come up with a coherent explanation. That's the biotic message. It was deliberately designed to resist evolutionary interpretations. That was the heart of the biotic message. The problem is it's not going to be accessible. Walter's book kind of proved that on some level, that
Mm-hmm.
the design is actually going to be a little bit hidden. It's discoverable, but it's like a hidden treasure. It takes work. So for my personal journey over the last 20 years or so, I've had to learn new bodies of knowledge that were painful to acquire, like
Mm-hmm.
chemistry and biology and a little bit more physics. But not that everyone has time for this, but if someone's really aching to see evidence of design, the opportunity is there. If God has endowed them with just enough ability to comprehend and learn some of this, they can learn it. And the more that I learned, it's like I could say every year I became more and more convinced of design. And at first I had some doubt that abiogenesis could happen, but that just got erased. And definitely now that I saw Change Tan and Rob Stadler's book, they just annihilated it. Did you see Rob Stadler's video on Standing for Truth?
Yeah, I watched both those lectures on the evolution book and the Stairway to Life. To me, it's almost like the onlooker says, that looks easy to do. If they're watching someone play ping pong, or watching a chef do a certain thing, or watching someone put up drywall even, there's a methodology to it that escapes, that it's not apparent, that a person can't just watch it and then do it. It's like the magic trick. You can't just look at the magic trick and say, this is, I could do that without knowing exactly how.
Well,
So
I think we can. I wanted to invite you to. I'm making the chemistry series for individuals that want to learn, and it might. I'm hoping that it'll plug a few holes. So, if
well, for me, it's all about how can I present information in a compelling way,
yeah,
so that it's an art form to present information. Once you have actually discovered all the existing patterns, now you have to put them in a way that actually matches the intake of patterns that the audience has.
well, I've gotten to the point. It's like maybe there's not the first off, the educational system has failed. The public educational system has failed. They're being shoved down irrelevant stuff. If they knew a little bit of basic math, they would start to understand some of this. If they were taught a little bit more basic chemistry, it would really help. And I feel like also, I felt a little embarrassed there when you asked me what's a covalent bond. I said, I ought to be able to answer this. And I couldn't. And it's like, yeah, Sal, this is why you need to relearn, even though I've had graduate level biochemistry. It's like, yeah, oh my goodness, sometimes you get thrown this basic question. It's like, oh my goodness, I need to be able to explain this succinctly. So where I was headed with that is, and I'm trying not to be too critical, but maybe I have to be, is this idea of just having debates after debates in these 1 hour presentations. That's like, I'm thinking time could be better spent. What if someone spent, instead of watching 10 debates where it's just the same recycled argument over and over again, both sides, why not spend 10 hours learning chemistry? So I'd like to invite you to go through my chemistry series that I'll be putting on the evidencereasons.org website. And before we met, I was actually working on the website to fine tune it. It'll also, God willing, be available on my YouTube channel that got reinstated. But we don't, as I had cautioned, I don't know how long it will last, given they're deplatforming people like crazy. And they deplatformed me for a couple days.
Now, do they deplatform because someone flags your channel?
They never explained what the process was. I think it could be a combination of automatic bots or whatever, plus user complaints, and then just maybe dysfunctionality in organization. I don't know, but that was a negative experience enough to warn me. I said, I don't want to do business with these people.
You don't want to have to rely upon them.
Exactly. They're very good for getting on YouTube, is a great way to be discovered by search engines.
Yeah.
But back to where I was headed, I'd like to invite you to go through the chemistry series. Now, even just like, if I would be grateful if you went through the first, say, 4 or 5. Now, how many is in a 2-semester chemistry series? I estimated they're going to be like 120 1-hour lectures with homework. So it's not for the faint of heart. And what I had intended to do is, after I've gone through this process, maybe I could extract the essentials and then make it more compact. But I've tried to say, okay, so let's say someone spends 3 days of their life, they learn the chemistry, they learn the arguments at the cell biology level, and then they go away feeling assured of design. I feel that would be worth it for someone's life, or even a week. And I look at what some of these Christians, they go off to these Christian colleges and they're spending all this time. And it's like, God, why don't they spend a semester learning the design argument, studying, really devoting themselves to hard topics and learning this? If they spent 16 weeks studying this than some other topic, they might be better off. It might cure their doubts. Because I hear these horror stories where these students go off to these even seminaries and they lose their faith. I'm just like, well, yeah, you know, there are arguments that are available to them that are not being taught. And because the material is technical, sometimes they think it's irrelevant. It's like, why should I study general chemistry to help me get closer to God? And that's what I'm trying to bridge. I'm trying to tell people, why don't you look in places that you'd least expect? Because maybe the cornerstone that you're rejecting is the very is the most important one, or one that God could really help you with if you're willing to invest time. And that's what I'm trying to persuade people is maybe take the risk and try. At the very least, you're going to feel better about yourself because you understand chemistry. Because now, like I was helping my mom deal with her urinary tract infection. I knew a little bit of biochemistry and cell biology. I was able to help look at the professional literature on treatments that were alternative to antibiotics. When the patient starts to develop antibiotic resistance, you're kind of forced, your hand is kind of forced to look at alternatives when the antibiotics lose their effectiveness. I happen to
Like
find
the natural healing processes of the body.
I found a very important, very simple substance that originated with a home cure, and it's a substance called D-mannose. It is a sugar that prevents bacteria from adhering to the urinary tract. And so even though it doesn't kill the bacteria, it gets flushed out rather than staying inside the patient. And so even we practice hygiene of just moving bacteria, not killing it, but just removing it. That's brushing our teeth every day. And it's kind of the same idea. We're not going to try to kill all the bacteria. We're just trying to get it moved out of the body. And is a very simple cure, and my mom's been recovering. So
that's the application of knowledge. It's like when you are discussing theological issues and then you're talking about origins, you're trying to bring together the words of God and the works of God.
Yes. And I've been reading the Psalms. What does it say so many times? I will declare the works of God. And that has more meaning now than any other time, because Jesus said, consider the lilies of the valley, how they grow. In the 21st century, considering the lilies of the valley, how they grow, has a totally different meaning, because we understand now, okay, it involves DNA replication, it involves DNA translation, it involves gene regulation, combinatorial gene regulation, a whole bunch of other processes. Now that we know it, and Jesus said, truly I say to you, Solomon in all his glory was not arrayed like one of these. That may have sounded like a figure of speech, but that's literally true, because we cannot build cells from scratch. And the majesty and the knowledge and the wisdom in a single cell of a plant is far beyond all the collective knowledge of all humanity, its sciences and engineering, by several orders of magnitude. If we look at the cell, if we're equipped with the chemical knowledge of cell, chemistry knowledge and cell biology knowledge, you will see that verse in a different light. And that's why I just don't like these short debates. It's like, guys, I wish you would just spend 10 hours studying chemistry, maybe 10 hours studying cell biology. You're going to see things in a different light. And maybe that's a time commitment, but that's certainly better than just going to modern day debate.
It's a cultural commitment,
Yeah.
I have a friend, Vishal Mangalwadi, who's trying to create a new educational movement using churches, where churches become bastions of academia like they used to be.
Yeah.
And
God
you
bless
have
them.
a cultural commitment to the Word of God and the works of God.
Praise God. That's the first time I've heard that. I'm glad someone has that vision. And it's really sad. It's the church leadership that is resisting that. I have offered to teach Sunday schools, volunteered to sit in, to explain intelligent design. I get cold shoulder. It's like, oh, that's great. We'll get back to you. And it never happens. Never happens.
I think it's primarily because right down to the foundation of our culture, we in the church community aren't embracing the elements that the Bible puts forth as, what we need. Like one of the elements was God told Israel you were to be a lender to nations, never a borrower, because the borrower is servant to the lender. And one of the problems with any culture is they have to be a going concern in some way. Now, the United States, when it started with 13 colonies, the Quakers of Pennsylvania started their own bank, and they started issuing their own money. And this gave them that engine of moving forward when they always had liquidity for everyone who wanted to work, could work. They put the highest level of existing potential to work through that. And it's because the church today lacks these fundamental elements in it. Like every church should be a bank to a certain degree, that like a public situation where they're issuing their own money. And if they can't do that, if they're not allowed to do that, then every church needs its own legal team so that they can then create the access to what they need on a fundamental level. And then once you have that, you're no longer dependent upon an outer system. You're now independent.
Oh yeah.
Now you have nothing to complain about. You have nothing to worry about. Now we start to implement all this science learning as a necessity because it has to be part of a growing model.
Yes, yes, absolutely. Now, it might, given how very specialized we are, it's very hard to decouple any group with complete isolation in terms of money. But the idea of the church being a source of education, we could teach college level stuff so much cheaper. And I see these college courses that are just absolute garbage, and a bachelor's degree at Duke University costing about $300,000 for a garbage diploma. And we could teach stuff that is so much more valuable for a lot lower price, and it's not happening. And yeah, so
the whole Western model's off. Like Japan, they were completely disenfranchised. They were sent to zero after Hiroshima and Nagasaki. And then they asked for help. How do we rebuild? And the person that came from the United States to teach them how to rebuild their economy basically spilled the beans on you need a public central bank, and you need to have a community sharing. That's why in Japan they have a patent pool where any new patent that comes in existence is owned by the government, and any company can dip into that pool and take advantage of that patent.
interesting.
So there's this homogeneous sharing of potential that's being created. But Japan perfected the public central bank, and 5 other countries copied that model: China, Taiwan, South Korea, India, and Singapore. And those are the 6 richest countries,
Interesting.
most advanced countries in the world. It's because they have the fundamentals right.
And it's really interesting that you're saying this, because for knowledge, it shouldn't, in the internet age, it shouldn't come at these outrageous prices that the universities are charging.
Yeah.
And what ended up happening is the public schools are not really public schools; they are scams that that plunder and pillage the public funds and don't deliver education. It's
They're
not
part of
really a
social
public
engineering.
school. Yeah, it's exactly. It's because the cost of actually teaching the kids shouldn't be as high as it is, and they're
It's
doing such a
because
lousy job.
when you don't own the goose that lays the golden egg, you have to rent everything you use.
Yeah.
But once you have that steady supply of gold, you're never at a loss for anything you need.
So, at this stage in my life, at least let's say just for over perhaps the next year, and God willing, nothing really adverse happens, and we never know because we're not promised tomorrow. But I'm committed to trying to make something publicly available to the creationist community. And my message is, guys, those debates are great for like the first or second introduction. But if you really want to get serious, you're going to have to learn some new ideas and learn the terms. And I'm going to try to provide the material. And it's just going to be that. I'm going to,
Yeah.
but maybe I could advertise it in terms of like, this is going to help you just as a person and a human being, that you know something you didn't before. And, you know, maybe now when you go to the store and you read product labels and all the ingredients, it'll be satisfying that you understand a little bit of what those terms mean. When you deal with a doctor and you're dealing with pharmaceuticals, you're learning what these terms mean. It may actually help you. It may save someone's life in your family or yourself, that you
Well,
just know a little bit more.
to me, it's like I finally got to the point this year. I finally given up on federalism. That it's just a nauseating mess that does not serve my interests in any way, shape, or form. I have to go back to localism. But the minute I consider localism, this tsunami of responsibility hits me.
Yeah.
Everything that I'm delegating to these centralized foreign bodies, I have to now take responsibility for myself.
Well, I think we can have another conversation, and I hope you'll. One way that you could benefit me is if you go through the chemistry course and you give me some feedback on it.
Mm-hmm.
I'm
Yeah, I
not
think that's what
expecting
you
to go
need.
to all. I'm first off, I'm not expecting anyone to go through all 120 lectures. I've only put out the first 3,
Mm-hmm.
but
You have to develop a format where you create a feedback loop,
yeah,
and you start tapping into the synergistic potential of the community you're building because
yeah,
of that, and start delegating parts of what you do to people who are maybe more talented in producing that element that you
yes,
need in order to convey an idea.
yes, and sometimes you just have to. It's kind of hard to steer a parked car. Sometimes you just have to move a little bit, and then you can get
Yeah.
pointed in the right direction. That's where I'm at right now
Mm-hmm.
with the websites. It's like I'm just gonna move and stumble forward and crawl, and hopefully I'll
The
be able
lessons
to
of the
walk.
doing.
Yeah,
Mm-hmm.
yeah.
For sure.
So I'm sorry I have to kind of bail out on you. We've been talking now for 11
Close to
o'clock.
3.
Yeah, close to 3 hours. So this is going to be a nice long video for my channel.
Cool.
And so thank you for the content. And it's been a great conversation. I just, as a heads up, I am backlogged.
Mm-hmm.
I have promises to other people for interviews and stuff. And because I had to take off a week to take care of mom, I'm even more backlogged. So I don't know when our next meeting will be, but you'll see me on the net and chatting. And
Mm-hmm.
chatting is a lot less stressful for me.
Yeah.
So we may run into each other that way. I want to really thank you for your interest in Biotic Message and the work of my dear friend Walter ReMine. So I'll leave you with the last words, Jeremy, if you had any closing thoughts.
Well, if we can't meet tomorrow, that means I have time to read the Stairway to Life. So that's basically what I need to do, in the interim, is to really approach it and react and see, how I have to start embarking on my own education, start looking up definitions and getting very familiar with the subject matter.
That sounds great. Well, I'd like to thank you for joining me. I'd like to thank all our viewers that have watched this video and heard our thoughts, and who probably hopefully got acquainted with the work of Walter ReMine. Take care, and God bless you until we meet again.
Okay,
Good
God
night.
bless.